Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Exploring the Range of Electron Microscopes, Resolution, Size, and Users. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, which is found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you also to submit questions as well via the comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank JEOL, who developed the content for this presentation. Since 1949, JEOL legacy has been one of outstanding innovation in developing instruments used to advance scientific research and technology. JEOL has over 70 years of expertise in the field of electron microscopy, more than 60 years in mass spectrometry and NMR spectroscopy, and more than 50 years of e-beam lithography leadership. Now it's my pleasure to introduce our speaker for today's event, and I'd like to introduce you to Alan Merck, electron microscopist with Leidos Biomedical Research, and there he is. Hi, Alan. Alan has spent 8 years as a microscopist in the lab of Srinam Subramaniam and at the Center for Cancer Research, NCI, developing methods for high-resolution data collection on a 300 kV Titan Krios. And Alan then joined Yana Ogayanovic's group at the Frederick National Laboratory, where he works on pushing resolution limits on 200 kV microscopes, including cryo-ERM 200. Now, it is my pleasure to pass the mic and the controls over to our speaker, Alan. So, Alan, when you are ready, you may begin.
Thank you very much for that kind introduction, Sonia. So my name is Alan Merck. I'm an electron microscopist at the Frederick National Lab for Cancer Research, and today I'm going to talk a bit about some of our experiences with the cryo-ERM 200. So just to give a bit of a brief background, because I know many people in the audience may not be too familiar with the structural biology field, so just to give a very brief history of the cryo-EM field and where it fits in the context of the full structural biology. So nowadays, cryo-EM is routinely used for high-resolution structure determination, but that wasn't always the case. So before about 10 years ago, cryo-EM used to be relegated to a low-resolution method, and in fact, the resolution of the structures were so low and the features were so poor that cryo-EM was actually called blobology. And that all changed about 10 years ago with the advent of direct electron detectors. So before the direct electron detectors were commercialized and given out to customers, the technology that was used was actually scintillator technology, where the electrons would strike the scintillator. That would be the electron signal would be transferred into photons, and it was actually the photons that were detected by the cameras. And as you can imagine, having this middleman in the process there really adds noise to the entire system. So back when it was the low resolution range, back before the direct detectors came online, you were stuck in the low resolution range, and you were lucky if you were able to get to, say, sub-nanometer resolution where you could see helices, and then if there was a crystal structure available, you could dock it in and try to make some biological interpretation. But once the direct detectors came online, the signal-to-noise ratio in the raw micrographs immediately improved, and that immediate improvement in the signal-to-noise and the micrographs led downstream to an improvement in the final resolution of the reconstruction. And once people got to the point where they could actually see side chains in their maps, that's when they were able to actually start building their own models de novo and depositing, more and more their own models into the protein data bank. So shown here in the center of the slide here is actually a figure from one of Richard Henderson's recent presentations, where he actually shows over time how the number of model depositions into the protein data bank changed over time. So as you can see from the plot here, before about 2000, cryo-EM had almost zero PDB depositions. They were orders of magnitude behind both NMR spectroscopy as well as X-ray crystallography. And then about 10 years ago, you see a huge inflection point in the cryo-EM graph where it just, goes off to the races. And now it's, as far as number of PDB depositions, it is past NMR, and it is rapidly approaching X-ray crystallography. And Richard, he's always, to his credit, he's always had so much faith in the method. I remember I was lucky enough one time in my career to go to a GRC, and I remember this talk about how Richard had said something about about world domination. And when he said something about world domination, he wasn't, talking about world domination for himself. He was talking about world domination for the cryo-EM method relative to the other methods. And this was about 10 years ago when I was there. And I remember thinking at the time that, even though cryo-EM was starting to get to high resolution, the possibility for cryo-EM to become the dominant structural biology method, seemed very, very unlikely. But Richard believed in it, and to his credit, it turns out that he actually, and this is mentioned in the piece by Owing shown at the bottom here, where it says the revolution will not be crystallized. Richard actually said this going back to even the 90s at a 1997 GRC, and he also kind of laid out the theoretical framework for why it was possible in this paper from 1995, which is shown on the right-hand side of the screen here. So in this paper from 1995, again, 1995 was, if you look at where, on the plot in the middle, where cryo-EM was as far as PDB depositions, the idea that somebody would come out and say that cryo-EM could be used for atomic resolution structure determination of small proteins. I wasn't in the field back then, but I can only imagine how crazy that must have seemed. But obviously Richard was able to see the future of it. So, if you're interested in the cryo-EM field, definitely this paper right here by Richard, this famous review, is definitely worth taking a look at. So before I started my work here at the Frederick National Lab, I used to work in the lab of Shriram Subramaniam at the NCI. And actually my first week when I got to Shriram's lab, it was actually the week that we got our first direct detector installed. And that direct detector was a Falcon 1 from Thermo Fisher FEI at the time. And it was the best detector that they'd ever had in the lab. It had the highest EQE. But when we started collecting on it, we saw an improvement compared to what we had before, but not quite enough to push us to atomic resolution or anything like that. And there was another direct detector that was out there at the time, a K2 from Gatan. And Shriram, based on everything he had seen in the field, he thought that actually the K2 was a better detector and a better way to go. So after about a year of us using the Falcon, Shriram made the switch to take the Falcon off the Krios and put the K2 on. And once we started collecting data with the K2, we had that immediate improvement in DQE, and then that immediate improvement in SNR on the micrographs led to an immediate improvement in the resolution of our structures. So whereas before on the CCD technology or with the other early direct detectors, we were getting to about sub-nanometer resolution. Once Shriram made the switch to the K2, immediately we actually saw an improvement in the map up to a resolution where we could fit in side chains. So after we got to, and the resolution we got to in that early map was 3.2 angstroms. After we got to 3.2, we were all excited in the lab. It was the first time we had ever seen side chains in the map. But Shriram, he was not satisfied, and we had a couple meetings where Shriram was adamant that we could push the resolution further. So we went back to the drawing board, went back to every step in the process. Even grid making, image processing, all that kind of stuff. We actually fully changed the grid making device that we were using. We made a switch there. That I think was a key difference, and also made differences in data collection strategies, image processing, even down to particle picking. So a lot of little changes I think added up to a big change where we went from 3.2 angstroms to 2.2, and that's what's shown here. So for us, it was the first time that we were able to see features on the side chains and also kind of on the backbone, carbonyls, that kind of thing. And nowadays, 2.2 angstroms by cryo-EM is routine, and certainly by X-ray crystallography standards, 2.2 angstroms is just kind of in the middle of the range. But at the time in the cryo-EM field, it was a super exciting result, and we were really happy to be able to see such fine features in our map. So when Shriram made the switch to kind of go start his biotech company in Vancouver, before he kind of fully moved up there full time, there was this transition period where he was still advising the NCI on equipment and that kind of thing. So when we were making the switch up here to the Frederick National Lab, Shriram made the decision for NCI to actually, when they were purchasing a microscope for the operation here, to go in a completely different direction, both as far as acceleration voltage and also vendor. So in Shriram's lab, we used the Krios, which ran at 300 kV, the Krios being from Thermo Fisher FEI. And then up here, Shriram made the call to go with a 200 kV scope and also to go with a scope from a new, different vendor than he had before, from JEOL. And Shriram, for various reasons, he was really excited. He really believed in the potential of the CryoARM 200 to produce high resolution structures. It was the first JEOL microscope that had this multi-specimen cryo holder. Shriram liked that, but the big thing that Shriram really liked was the fact that this was the first cryo microscope where you could have a cold feg. And Shriram, he thought that all the kind of the technical challenges of using the cold feg during data collection, the flashing and all that kind of thing, he thought those were worked out enough that the potential theoretical benefits of the cold feg could actually be shown to have a practical benefit if we were able to get this microscope in and do a good job with it. So, the goal was to use this new microscope, lower kV, with this brand new kind of amazing tech with the cold feg to be able to produce densities that are as good as what we would get on a 300 kV scope. So after we had placed the order for our CryoARM 200, we were waiting for it to get delivered and installed and that kind of thing. And there was actually already results coming out from other CryoARM 300s around the world. So other cryoarms around the world. So shown here is a CryoARM 300 structure on a protein called apoferritin. And this has become a standard benchmarking specimen in the field. But even with that, when they posted this structure of apoferritin at 1.54 angstroms, it was the world record for the highest resolution. And so we were, again, sitting here waiting for our CryoARM 200 to get delivered, and we were very excited about the CryoARM 200. But when we saw this result of the world record for the highest resolution, our excitement only increased, an immense amount. So when we eventually did get our CryoARM 200 installed, because we were collaborating with Shriram at the time, and Shriram in his lab, the benchmarking specimen was always beta-gal. So because we were collaborating with him, we used beta-gal again, and we would kind of apply the same process to beta-gal on the CryoARM that we had done on the Cryos. And this was one of our early structures here where we were able to get that same protein, which was at 2.2 angstroms before. Now we were able to get it to higher resolution. And a lot of the features, you see some of the same features as far as holes in aromatic rings, water molecules, that kind of thing. But what we see that's new here is we actually start to consistently see bumps for the hydrogen atoms in the protein. And here's a bit of more of a comparison between the Cryos K2 structure and the CryoARM 300. So the Cryos K2 is boxed in red. The CryoARM K3 is boxed in black. And as you can see, everything is just a little bit sharper, a little bit more crisp. The one thing you'll notice, though, is that around some of the atoms, you start to see, around some of the atoms on the model, you start to see these bumps in the density. And when we first saw that, we weren't really sure how to interpret it. And we thought, we didn't think it was noise because we thought the map was really good quality. And, there's obviously different ways to interpret it. But lucky for us, there was a paper that was released shortly beforehand by Dmitry Lumkis's group, where they reported the 1.86 angstrom structure of adeno-associated virus. And in that 1.86 angstrom structure, they, for the first time in the history of cryo-EM, were actually able to see hydrogen atoms in density for hydrogen atoms in their map. They saw like these bumps coming off the side chains. And, so after we started seeing those consistently in our map, and then we recalled what Dmitry had published, we put two and two together and we realized, oh, that's what they are. Because when we were first seeing it, again, we weren't sure whether it was noise or potentially coming off of the parent atoms themselves. But once we saw the location of them and the quality of them, again, lucky for us that Dmitry had already seen these, we kind of knew what they were. And an example of kind of the confusion is shown on the right-hand side of the screen here. When you look at the lysine 621, the kind of the density bumps around the carbons, you could very easily say to yourself that we're kind of on the verge of atomic resolution as far as being able to separate the carbons from each other. But that wasn't the case. And if you look at how the density falls, you can kind of see that the bumps themselves are actually kind of only leading in one direction, i.e. the direction where the hydrogens are. So as far as the practical benefits of hydrogen, so getting to 1.8 angstroms at 200 kV was a very interesting technical result, and also being able to see the hydrogens, for me, the first time in my career I could see it, it was really exciting. But the question is, what's kind of the practical benefit? So when you do cryo-EM or you do X-ray crystallography, obviously the ultimate goal is to use the map that you have from the experimental method to be able to fit a model in, and the better resolution the map is, the more precisely you can fit the model, and the less kind of flexibility there is to move around, the more you're confident on the position of the atomic model. So with the atomic model, one of the very difficult parts of fitting the atomic model in crystallography at a similar resolution range is that the asparagines and the glutamines, there's ambiguity in fitting of the amide group. So what we're showing here is in cryo, where you're able to see the hydrogens at 1.8 angstrom resolution, which you couldn't do in X-ray crystallography at a similar resolution, you can actually use that extra density from the hydrogens to confidently fit the amide groups. So, and in fact, it's such a well-known problem in structural biology. It's called the NQ problem. that there's actually programs that are specifically designed in X-ray crystallography to go through the map and to find the NQ rotamers which have been fitted incorrectly and to flip them to the right rotamer. And it's estimated that about 20% of the NQ rotamers are fitted incorrectly in X-ray crystallography maps. And usually with X-ray, the reason they're able to get to the fit to kind of beyond the 50-50% is because what you do is you look at the surrounding chemical environment, and you have one hydrogen bond donor, one hydrogen bond acceptor, and you say, okay, I can fit based on the surrounding environment. And for many times, that's actually accurate enough to do it properly. But still, there's 20% of the time where there's not enough information there for you. So having this extra information that's kind of completely orthogonal to looking at the surrounding chemical environment allows you to fit these in more accurately. So if you're confident in your, in density for the hydrogens, then you, at least in principle, you could actually fit the amide groups purely based on visual inspection. And so that's what's shown on the left-hand side. And then on the right-hand side, you kind of see a similar thing for the threonines, where now because you have the extra density coming from the hydrogens of the methyl group, there's no longer ambiguity in how you fit a threonine. And now here's just kind of, the 20 standard amino acids, one representative density for each coming from the 1.8 angstrom beta-gal map. So after we had gotten this map from 200 kV cryo-EM, we were really excited about it. People in the field, they were happy to see that this result was here, but naturally it led to some questions. And I've listed a couple of the questions on the bottom right here that I'll try to answer in the rest of my slides. So the first question that people would ask is, okay, 1.8 angstroms is high resolution for 200 kV, but it's far from the world record. So is 1.8 angstroms the limit for this type of microscope? So that was the first question that people would ask. Another question that people would ask is beta-gal. So beta-gal was actually done, it was, again, Shriram's lab kind of standard benchmarking specimen, and he had been working on that ever since he worked with Richard at the MRC 20 years ago. And as far as I know, the idea there was that back then when you had this technology where the hardware and everything else was limited, you wanted to choose a particle that A, had reasonable symmetry, beta-gal's T2 symmetry, but also that's a large enough size that you would have even a fighting chance of aligning it with the technology at the time, or even seeing it in the micrograph. So beta-gal it's not huge, it's 460 kilodaltons, but it's large, relative to the average size of the protein in the cell. So if we, really want to use the CryoARM 200 to get some biological insights on a wide range of samples, obviously it's got to go far beyond 460 kilodaltons. And then the other question that people were getting a lot was how this microscope would perform on biologically relevant samples. And when people would ask this, sometimes they would even come out and say, how would it perform on a real sample, right? Because beta-gal, again, it's been done by it's been done to high resolution by cryo-EM many times, and it's obviously been done to high resolution by X-ray many times, and by all different groups, that kind of thing. So beta-gal is it's a very, very nice standard specimen, but it's not and it's from E. coli, the one that we use, so it's got no relevance to cancer, which we work on here. So one question that people have was how this microscope would perform on a biologically relevant sample, and when they would ask that, what they would kind of really be asking us, so they want to know how this microscope would perform on a biologically relevant sample, but what they're really thinking is, okay, how would this microscope perform on my sample? So we were excited to answer that question. So in order to answer that question about how this microscope would perform on someone, a sample that people have a bit more of a kind of a relationship with, but Jana said, okay, what's the sample that we can think of that the most people could relate to? So Jana was like, okay, there's nothing, no specimen has been done more often in cryo-EM than the ribosome. So she goes, okay, let's do the ribosome. But in addition to that, let's choose the most complicated ribosome, which is the human ribosome. But then furthermore, Jana said, okay, to really stack the odds against us, let's not try to lock the ribosome in one state. Let's just take ADS particles from HeLa cells, put them right in the grid, image them, and see how it goes, and do the best we can to manage this heterogeneity and see how it goes. So that's what she did here. So Jana, again, purified it straight from HeLa cells, the ADS unlocked in any state, so in all of its heterogeneous glory. And to deal with this heterogeneity, obviously she had to do the usual stuff as far as 3D classification, that kind of thing. But in addition to that, because of some of the flexibility between subunits and inter-subunit for the small subunit, she had to use multi-body refinement in RELION in order to eventually get it to high resolution. So you can see on the bottom left, you can see the masks that Jana used for multi-body refinement. The blue mask around the large subunit, and the dark yellow and light yellow masks around the small subunit, body domain and head domain. So after all this image processing and everything, Jana was able to get the global resolution of the ADS to 2.1 angstroms, which is a very exciting result. But in addition to that, in addition to the fact that it's 2.1 angstroms globally, if you look at the local resolution plot on the bottom in the middle, where the map is colored by local resolution, you can see in the core of the large subunit that the resolution in the core actually extends to 1.8 angstroms, which is the same resolution that we had beta-gal at. So it was, that confirmation for us that our cryo-EM 200 is not a one-trick pony. It can do more than beta-gal, and in fact, it can do a particle that's been done many times in the field. And because it's the ribosome, obviously you have, when you look at the map, you see so many different things than you would see with beta-gal. So just shown here is some Watson-Crick base pairing, both canonical and non-canonical. So now here you just have the 20 standard amino acids from the ribosome map, like we had for beta-gal. And then again, in addition to that, because it's a ribosome, you also have the RNA, and that's shown on the bottom right. And again, with the ribosome, because it's so big and complex and so many things going on, you have a lot more to look at and to sort through and to model. And obviously, the metals and the solvent overall is key to ribosome structure, translation, fidelity, and speed, and all that kind of stuff. So it's quite a chore actually to go through all the solvent and to model it correctly, just purely on the number of different things that you have to go in and model, and also sometimes it's not so easy. You have to really take in a lot of different information when you decide to assign the solvent density. So, but getting to the higher resolution does help you because, for instance, with the magnesium, with kind of this classical octahedral coordination, you can really make out the distinguishing between the magnesium and the water. And it's for a lot of those, some of those it gets easier, but there are also challenges when you get to higher resolution as you see more things. As we kind of talked about earlier with the hydrogens, that kind of thing. But so you just shown here as examples of magnesium, zinc. The zincs are trivial. The potassiums are ultra difficult, I think, and distinguishing potassiums between waters, and there's some different ways to do that as far as how they coordinate in waters and that kind of thing. But the potassium is out of the one shown here, the potassium is probably the most challenging. And then shown on the right, again, the ribosome, complex, and especially the human ribosome, complex in pretty much every way possible. You have these post-transcriptional modifications, which there are more of in the human ribosome than in lower organisms. So in the higher resolution maps, you can actually start to visualize those chemical modifications directly in the map. And that's shown here for the post-transcriptional modifications. And it's also shown here for the post-translational modifications. So the one on the right, you can see a histidine. That's a post-translational modification on a side chain. That's kind of what I think a lot of people think about when they think about post-translational modifications, but also in the center, you have a backbone modification of this isoaspartate, which you could see in the small subunit map. Again, just to make things relatable and also to kind of increase the challenge a little bit each time, Jana then decided to say, okay, another relatable thing would be to go after something related to COVID. So what Jana's done here is non-structural protein 1 from SARS-CoV-2 bound to the 40S ribosome. And it turns out that you have, at least in our hands, and I think in most people's hands, doing just the small subunit is much more difficult than doing the 80S ribosome or a 60S. So it was a bit challenging to get this all to work, but obviously she was able to pull it off. And as we get to higher resolution, we can actually see that the way that the NSP1 interacts with the ribosome, there's obviously plenty of direct interactions, but there's also a key interaction that's mediated by a water molecule, which we were able to actually see in the map once we got to about 2 angstrom resolution. So going back to some of the questions that people in the field had asked us about the microscope and the performance, again, 1.8 was an exciting result, especially since we got it early on. But then the question was, okay, is 1.8 angstroms the limit for this microscope at 200 kV? And I'm happy to say that it for sure is not the limit. So, Jana came to me and she's like, okay, 1.8 is great, but we got to at least try to push this. We knew that the 1.8 was not the limit. We knew that there was a lot of things that we can do better. So Jana went back all the way to the biochemistry to change the protein purification, everything like that. We changed a few things here in the data collection and even made some adjustments in the image processing. And again, those a couple of those small improvements kind of, the cumulative effect was added up to kind of a relatively large improvement, at least in this resolution range, where we were able to go from 1.8 angstroms to 1.6 angstroms. And we'll just kind of see what it looks like in the movie here. And it looks very similar to what we would see at 1.8. Everything just kind of looks a little bit sharper. You see a few more of the hydrogens, they look a little bit sharper. A couple of the extra features that you can see in the top left-hand corner. Now you start to see the second hole for the tryptophan more often. And also, again, the hydrogens on there. And there's a couple other features that you start to see a bit sharper at 1.6 than you did at 1.8 for the same sample. So it was exciting to get to such a high resolution on beta-gal, but obviously if we can only do it on beta-gal, it's meaningless to the world. So we were excited to try to do some of the similar higher resolution stuff on more meaningful samples. And for that, Jana decided to go with the large subunit of the human ribosome bound to the FDA-approved drug ombutaxine. And the idea was obviously to see what the resolution can do as far as seeing a density for a drug and how that fits and how maybe you could understand the interactions better. And also, just to, again, kind of prove the point that beta-gal is not the one-trick pony that this thing can do, and do something that's more relatable to people. So we got it to overall, the number here is 1.7. It's a bit beyond that. It rounds to 1.7. I forget actually what the exact number is. But even though the global resolution is about 1.7 angstroms, in the core of the particle, on the left-hand side, you can see where it's colored by local resolution. In the core of the particle, the resolution actually extends to slightly beyond 1.5. And even on the periphery of the molecule, you can see on the outside where it's colored red, but that red is 1.9 angstroms. So across the entire span of the map, it's beyond 2. But when you look in the core, where it's 1.5, it's a beautiful, beautiful density. So the ones here shown for the drug they're towards the middle. I think they're not quite the core. So if I had to put a number on it, maybe they're about 1.7, around there. But then for the core of the particle, it is where it's kind of inching closer to 1.5, then we really start to see some nice density. And that's shown here both for protein, RNA, post-translational modifications, post-transcription modifications, all that kind of thing. And again, here you start to see some of the differences between at least cryo and X-ray is that, so if I look at the arginine on the bottom right, it looks like the map really follows the carbon atoms really well up the arginine. So everything looks good there. You say, okay, we see those bumps. They're probably for the heavy atom there. But then if you look at the arginine in the middle, which is next to that proline, at least based on that fit right there, it looks like there's only the protrusion coming from the one side where the hydrogens would be. So, we talk about this all the time, exactly how to interpret this and whether with the fitting you take into account the hydrogens for the side chains and all that kind of stuff. And obviously for the arginine in the middle, in the center, if you try to fit those individual carbons into the center of the density, you would end up with a non-physiologic distance, right? So to kind of just break the bond. So we know that for that one, we know it's not right. And, but for other ones, it's not so clear, especially in the core of the particle, you're at beyond 1.5. So you're actually, at least in theory, you're at a carbon-carbon bond is, longer than that. So you actually have the potential to be able to separate atoms, but we're not there yet, even in the core. But, but it does get confusing about exactly how you interpret this density. And it's something that, again, it doesn't exist in crystallography. That problem doesn't exist in crystallography because in crystallography, at this resolution range, you wouldn't see contribution from the hydrogens. I just want to show a couple of movies here. So on the left, you see ribosomal proteins, and on the right, you see the RNA along with solvent, both water molecules and magnesiums, which get a little bit of everything there. So after we had the high resolution structure of the ribosome, Jana decided to go in and use Servalcat from Garrett Mershadov to do difference maps to try to pull out not just these these large densities for the hydrogens, but now with the difference maps you can actually see punctate spherical densities for the individual hydrogen atoms. And the idea there is that you do your modeling however you do it, either with the hydrogens, but if you have hydrogens on your model, you delete those hydrogens from the model, and then you have that fitted into the map, and then you subtract the model from the map. And that difference, because the model doesn't have the hydrogens and the map presumably does, whatever positive difference peaks that are there, those are the hydrogens. And the positive difference peaks here are obviously shown in green. And you can see that, exactly where the hydrogens should be, you see a lot of spherical densities for the hydrogen atoms shown there along the bases and also along the side chains and, quite nicely along the backbone. The other thing that's shown here on the bottom right is, so when Jana did the difference map here, she didn't have the hydrogens in the model, but she also left out the PTMs from the model, the post-transcriptional modifications and the post-translational modifications. So, and the idea there was that, just to kind of have this sanity check where, because we know that the methylation is there on that particular RNA, we know the methylation is there. We can actually see it, of course, in even in the FO map. The difference peak from that particular density should be larger than the difference peak for just the hydrogen. And it turns out that's exactly the case. So it was a good sanity check, and it's just a good check to make sure that your process is working and that you're not over-interpreting anything. So again, like we talked about earlier with beta-gal, where you could start to see the hydrogens and you actually use that for the modeling. The question here is that now that you're seeing not just hydrogen blobs, but you're actually seeing stronger hydrogen peaks, where you may not have been able to see them before, can that help you in the modeling? And it turns out that we think that it has the potential to be a critical help. So as I talked about a bit earlier, with the ribosome, modeling the solvent is non-trivial. And any more information that you can get to increase your accuracy is very, very welcome. So what we're showing here is that two environments that are kind of at first glance they look very, very similar. However, when you look at the difference maps peaks, the solvent particle on the left has the two difference maps peaks corresponding to hydrogens, whereas the one on the right does not have those. And the question about the fitting of these two here is whether that solvent particle is a water molecule or a potassium. And again, sometimes you can look around the environment and figure it out, and other times you can't. But if you have the hydrogen atoms, if you can visualize those, then you know for sure that that's a water molecule. So the one on the left that has the two hydrogen atoms, you know for sure that that's a water molecule. Whereas the one on the right, which does not have the hydrogen atoms, we have a higher level of confidence that that's not a water molecule and that is potassium instead. And it turns out that with these two particular cases, Jana was already able to tell, based on the surrounding environment, which that the one on the left was a water and the one on the right was a potassium. So she was able to identify them even before we had the difference mass peaks. But so we have this kind of orthogonal verification again, where you kind of know what it is already. But it's again a good sanity check that the process is working and that you could potentially use this to correctly identify solvent particles as you get the higher resolution. And in fact, you can actually see it in the figure here. In the image on the left, you can see at the top of that figure, there's a magnesium there, and the water is coordinating it, right? So that the solvent particle is coordinating it, so you know for sure that that's a water and not a potassium. And then the one on the right, if we were in COOP, we could look around and figure out that it's highly coordinated and potassium, that kind of thing. But the other thing I'll mention is that for the hydrogens on the water molecules, we can see them in the one on the left here, but those hydrogens in the maps that we have, the hydrogens on the water molecules are by far the most difficult ones to see, and there's a lot of potential reasons why that could be. We have a couple, theories and kind of ideas about why that might be the case, but being able to see the hydrogens on the water is actually very rare, even in a map that's as high as resolution as the ribosome or beta-gal that we showed today. And as a kind of an example of that, if you look at in the screenshot on the left, if you look to the right of the magnesium, the water on the right actually does not show the hydrogens, even though it's right there with next to the water molecule that does show the hydrogen. So the general way that it seems to work out is that the hydrogens along the backbone, either the RNA backbone or the protein backbone, those show up first. As you kind of progress to get the high resolution, the ones on the backbone show up first, and then the ones on the side chains and the bases show up. And then if you're lucky enough to get to even higher resolution, only then do the hydrogens on the water molecules begin to show up. So just to give a bit more of an overview, just kind of the additional amount of information that you get as you get to higher and higher resolution. So shown here is a kind of a classic ribbon representation showing the large unit and the small unit, so kind of like this skeleton. And then as we get to high resolution, we could add more information on. So as we get to about 3 angstroms, we can start to see post-transcriptional modifications shown here in blue. As we get to a little bit higher resolution, we can start to see magnesiums and other metals shown here in green, which you can see a bit on the left. As we get to around 2.5, we can consistently model in the water shown here in red, and then as we get to hopefully beyond 2, then we can show the hydrogen shown here in cyan. So now you kind of see it when you start with that bare skeleton where you just have the sequencing, just kind of have the register down. As you get to higher in resolution, it just becomes completely decorated with much more information than we kind of had at the start. Again, going back to the fitting of the amides, obviously with the lower resolution beta-gal map and without the difference maps, we can see this bulk density for the hydrogens, and we use that for the fitting. But obviously now with the difference maps, now what was a bulk density now is 2 punctate densities for the hydrogens. We see that both for the glutamines and the asparaginines, and then similarly for the methyls, we can actually start to see punctate densities for the individual hydrogen atoms, and the fitting just becomes easier. And a few more shots of beta-gal here. As you can see, the side chains, which were just the FO map before, now they're completely decorated in hydrogens, and you see hydrogens along the backbone, beautiful ones along the side chains, and even the hydrogen bonding here with the tyrosine on the top right, hydrogen bonding to the carbonyl backbone. So again, going back earlier to some of the questions that people in the field were asking us, it was that, beta-gal was a very nice thing to do, but can you do it on a more biologically relevant protein? That was the ribosome. But with the ribosome, it's a more interesting one to do. It's something that's more relatable. But the problem is it's actually bigger than beta-gal, right? So in theory, it's potentially easier. So, we understood the question as far as whether we could use this microscope to push the resolution on things that were a bit more challenging, a bit smaller. And for that, we were, one of the proteins that we're working on is this protein shown here. It's 145 kilodaltons. The resolution that we currently have is 1.8 angstroms. And that's actually the map that's shown here on the right, where in the 1.8 angstrom map, we can start to see hydrogens again in the difference map. Now, for this map right here, we see hydrogens pretty well along the backbone, but along the side chain, they're much weaker. And, so you can see it a little bit on the phenylalanine in the bottom right, but for the most part, the side chains, you might see one for the beta carbon. And then as you go further up, it's a bit more flexible up there. You start to see them less often. So at 1.8, this is what you see. And the one thing that we were really happy about with this is that, so 1.8 is the highest resolution we have for the sample. That's the map that we're showing the screenshots of here. But we have multiple different data sets of this in kind of various conditions and things. And for all of those data sets, they all go to about 2 angstrom resolution. So it wasn't a one-off thing. It was actually, for this sample, everything goes consistently to about 2 angstrom resolution, plus or minus. So it was a very encouraging result. We say, okay, for 150 kilodaltons, we can go consistently to 2. And let's hope that we can, A, push the resolution further for this, which we're obviously currently working on, but also B, the goal is obviously to be able to do this for ever smaller proteins that are closer to the average protein size in the cell. So to go after kind of that, as far as smaller proteins, we tried this protein here, which is another protein that I think people have a lot of hands-on experience with, HSA. So, HSA, BSA. And this protein is 67 kilodaltons, so it's definitely on the smaller range of what can be done with cryo. And right now, the resolution we have is 2.5 angstroms. And the goal going in, and still the goal, is to get it to below 2 angstroms. Obviously, we're very well familiar with this work. There's very good work by Basil, Greber, and Abikateka et al. With 85 kilodalton protein, where A, they were able to get it to beyond 2 angstroms, but B, they were able to consistently get it to be that performance, right? So that's our goal here. And, we talk in the lab about what the resolution limiting factor is for this particular sample. And the consensus in our group is that the microscope is not the limiting factor, right? I think that we've proven that. So I think that we've kind of settled on the resolution limiting factor being the flexibility of the protein. Obviously, there's other things that we can do with, better grid making and that kind of thing. And, we're exploring things in the processing to get rid of some of the conformational heterogeneity. But obviously, the problem is, it limits your resolution a bit. So I think that, for right now it's at 2.5, but we have every expectation that it's going to go higher than it is right now, and hopefully we'll be able to get it to sub 2 angstroms before too long. But for right now, it's stuck at 2.5. So just to kind of sum up a bit about the 3 main categories that we had kind of looked at here as far as samples go. So on the left, we show kind of a standard sample of beta-gal, and that goes to 1.6. But again, beta-gal, very easy to produce, very easy to work with. And then in the center, we show the ribosome. And actually, what's shown on this side is something that wasn't shown in the previous slides, is that all the previous slides showed human ribosome, but here it's actually showing other organisms. So we have the human ribosome, the rabbit ribosome, and the bacterial ribosome, all at better than 2 angstrom resolution. So, it doesn't matter what organism you're working on, as long as you, do the biochemistry right and everything like that, then you're going to end up, you collect your data right on the CryoARM, you're going to get a good resolution. The other thing is that with the ribosome, and also with any specimen, if, one of the concerns is that you do your best to potentially try to lock the particle into one shape, but with the ribosome, that's not always possible, and there's always kind of unforeseen things. And it turns out that if you have this conformational heterogeneity, obviously nowadays with the different computational methods, there's various ways to still get the high resolution. But with the ribosome itself, if you have a bunch of different states, you can still get to obviously high resolution, but for multiple states. So just as an example, with one of the data sets here, it was a data set of either 10 or 15,000 micrographs. So not that large by today's standards. So 10 or 15,000 micrographs, and you can get to multiple high resolution structures, but actually 3 different structures from the same data set that are all beyond 2 angstroms. And you could see different things in them. You can see different conformations for things that are actually biologically relevant. So if, you do the best you can to stack the odds in your favor, but sometimes it's not possible to lock it into one state. And also, sometimes for the biological question you're going after, you don't want to lock it into one state. You want to allow it to do its thing like it would do in the cell. So definitely it's possible. And if you have a good enough data set, and if you have a high enough SNR in your micrographs, then that actually is not, it doesn't preclude you from getting to high resolution to the point where you can actually get to the hydrogen resolution like we showed in a couple of these. So another thing I should mention is that all the data that I showed today was all from, all data collected with CryoARM, and CryoARM was set up for us by applications engineer from JEOL named Taku Fugamura, and shown here on the right is me pictured with Taku in front of our CryoARM here. And I remember when I heard that we were getting the JEOL microscope for here, I remember I was so nervous because my whole career I had only worked on Thermo Fisher FEI microscopes, and with the Thermo Fisher FEI microscopes, the software, like the user interface on the microscope is all similar. So if you know how to work one, you kind of know how to work all of them. So I worked mostly on the Cryos, but I also spent some time on the Polara, T12, that kind of thing. But when I heard that I was working on JEOL, I kind of remember what people had told me about working on JEOL and how it was so difficult and so different from working on a Thermo Fisher microscope. I was very, very nervous going in that I wasn't going to be able to do a good job. And thanks to the software on the JEOL microscope, I think is actually pretty user-friendly, to be honest. I was much, maybe relative to what I was expecting, but I think it's actually pretty user-friendly. And the other thing is that the way that Taku set up CryoARM for us and the way that data collection is done here, the transition was really seamless. So, and to be able to use the software for data collection and to use it to produce high resolution data, there's really no doubt. And if I can do it, then anybody can do it. The other thing I'll say as far as other data collection programs is that everyone has their own preferences as far as which program to use. So some people prefer to use Latitude for data collection. And if you are one of those people, I'm happy to say that Latitude does work on the CryoARM. So we actually, a couple weeks ago, got the new software installed on the microscope side to be able to allow Latitude to do the specimen exchange automatically, to do this automated screening like people are increasingly doing nowadays. And from what I understand, that entire thing is already set up on other CryoARMs as well. So if you are one of those people who prefers to work on Latitude, and that's the only one you're comfortable with, you'll be able to use that in the CryoARM as well. Another thing, before I close, another thing I should mention is that the CryoARM that we have here is the first generation of the CryoARM 200. It's, I guess, the CryoARM 201, but JEOL has released a CryoARM 202. So depending upon if you're going to a CryoARM 200 for data collection, you may get one or the other, and I guess you could ask. And I don't know how many of these are out in the wild, the CryoARM 202, but there's, as with any change from a G1 to G2 or anything like that, there's some incremental changes, improvements. So one of them with the CryoARM 202 is that you have a better vacuum in the gun chamber so that you have to flash the tip less often. So I think when they first came out with the CryoARM version 2, JEOL recommended that you only have to flash half as often, but I think now, to be on the safe side, I think they've adjusted that a bit, where it's 1.5 full difference. So if you flash the tip every 4 hours before, now with the new CryoARM, you only have to flash it every 6 hours. And those are all, at least to me, they're good changes. Obviously, every incremental improvement is good, but out of all the changes that exist between the CryoARM 201 and 2, the one that I love the most, the one that excites me the most, is actually the switch in the objective lens to having a narrow gap pull piece. So the CryoARM that is behind me and the CryoARM that we use for all this data collection here has a wide gap pull piece. And the CC value there, the nominal CC value is 2.7. With the new CryoARM, the CC value is now 1.8. So, and after relativistic correction, you get the numbers on the right. So you get 3.1 for the one behind me, and then 2.1 for the new one. So, and just to plug those numbers into the focal spread equation that you see on the right-hand side, one of the assumptions that people sometimes make is that the contribution from high tension instability and from lens instability is much, much less than the contribution from energy spread. So you can kind of reduce that focal spread equation down to just C sub C times delta E over E. So your delta E and your E, you plug in 200 kilo electron volts, and delta E might be 0.3. And then you just plug in the CC value that you have depending upon which CryoARM you're going to. And actually, there was an interesting conversation on Twitter a while ago between Rado Donev and Wim Hagen, where Rado had said something to this effect about the delta E over E being the dominant effect, so you can, for the most part, roughly ignore what's going on with the high tension instability and lens instability. And Wim recommended that you don't ignore that. He said, don't take that for granted. And Wim didn't give any information about why he thought that, but obviously Wim has a lot of experience, so it was something in his experience on all the microscopes that he's worked on over the years that led him to say to not take that for granted. But obviously you can see from the equation that it turns out that even if you leave in the contributions from high tension instability and lens instability, even then having that CC value at the front multiplied by everything, whatever you had before, if you, all things being equal, if you drop the CC from 3 to 2, you get a 1/3 fold improvement in your overall focal spread. So it's it's something that potentially, especially in the high resolution range, it's gonna matter at some point. For us, we don't know whether it's limiting us yet, and to be honest, I don't think it is. But at some point, having that focal spread there is going to be a limitation, and having this this better CC value and also the better CS value too. If you're one of the ones that if you think that spatial coherence is limiting you as well, that could be a factor as well. But definitely with the focal spread, it could be a relevant factor, and so definitely all these improvements, and there's all trade-offs involved as far as tilting and a lot of nowadays funky microscopes going out into the field, especially if you think about Holger, and he has kind of a situation where he has with his CS corrector you add on CC, but then with the monochromator you drop the delta E, so it counterbalances that a bit. And it'll be interesting, at least for me, as I watch all these go out, it'll be interesting to me how they perform and how they all compare, and for the different use cases, what ends up being the best one. But certainly for us, with the applications that we've done thus far, for all the structures I showed today, if we had a narrow gap in the pole piece, things only could have been better. And it's just a question of whether that is limiting us, and time will tell. So just to close, as far as making things more user-friendly, we always try to do things in a way that allows us to have better information about the microscope. So one of the things that we've put together here is a Streamlit app where all of the microscope parameters that show up on the different vacuum menus, temperature menus, all those kinds of things, this Streamlit app will actually allow you to look at that and to look at the trends over time. So you have what is showing here, you have a menu on the left-hand side where you have the vacuum levels, the lens values, the reflective values, the apertures, all that kind of stuff. And then you can pull up that information and look how it is right now and look how it changed over time. And, so one of those things is that if you notice that you have the vacuum at a worse value than you had before, one question that you sometimes have is that when that problem started. And with this app right here, you'll be able to put it on your CryoARM microscope and just look at the values and see when over time the vacuum started degrading and, getting to a worse value, or any of the other values that you notice that are strange. Obviously, sometimes things go wrong and you don't really know that they're wrong. And with this, you'll be able to A, look at when they went wrong, and B, you can set different ranges where you kind of find them acceptable. So if they go outside a certain range, you'll actually automatically get an alert up in there. And then similar to that is actually a similar thing to this desktop, something that can run on the CryoARM computer, but now it'll run on your phone. And the main advantage here that I like is that it's set up so that if any of the values go out of range, so that the ones here in green are in range, if any of the values go out of range, then you'll get an alert on your phone. So that means if you're home for the night, if you're home for the weekend or something like that, and something goes wrong with the microscope where there's a vacuum crash, or if the high tension shuts off, you'll get an alert automatically on your phone, and then you'll be aware of it, and then you'll be able to potentially come here and recover the problem before it turns obviously into a bigger problem. Especially the thing I always worry about is a vacuum crash and the column is cold and that kind of thing. So with this app, you can it's set up so that you can get an alert on your phone. And the app right now, the one that I just showed in the video here, actually it was running on my phone. I have an iPhone, but we also have an Android version. Right now the Android version is only running on our laptop with in the emulator with Android Studio, but that one will run on the phone as well. So, and both of these pieces of microscope monitoring will be the source code will be given out. Anybody that wants to run it can run it, and obviously people, especially with microscope information, they like to keep that in house, and so you'll have the ability to do that. But also, if you prefer it, we'll be able to manage it for you as well. So if anybody has interest in this, then definitely get in touch with someone from JEOL or myself, and we'll make sure that it can get out to everyone, and everyone can monitor their microscopes and not worry about that. So just like to thank my team here. So shown in the top left is my boss, Yana Ogdenovich. I call her world's best boss, like Michael Scott from The Office. Shown in the next to her on the top right is Bernard Hyman. Bernard Hyman obviously been in the field for a long time, working on Vsoft and various other things, and we always love doing some crazy experiments with him so we can work to kind of understand some of the fundamental theoretical things about cryo-EM, which we never even thought about before. Shown on the bottom right is Daniel Cleary. He's someone that's relatively new to the group and to the field. He never worked in cryo-EM before, but he's our machine learning AI expert, and he's catching up to speed on cryo-EM as well. And then shown on the right is Kendra Lee, and shown on the left is Dennis Winston. And they produce samples in the lab, but also they have quite a bit of computational expertise as well. So with that, Sonia, I'll send it back to you, and I'm happy to answer any questions anybody has.
To send in your questions right now by using that questions window, and we'll try to attend to your questions during the time we have together. And for those of you watching on LinkedIn Live events, feel free to post your questions in the comments section. Okay, so let's start with this first question here. This audience member is asking, Alan, how much is radiation damage limiting the resolution?
It's a very good question, and we think there's been obviously work done on this before, and every data set is a little bit different as far as the pattern of the radiation damage and the amount of beam-induced motion that you get from frame to frame, and even within a data set, obviously every image has the potential to be different, but it definitely is likely a factor, and also something that there's been some work from the MRC about looking at the zero dose maps, kind of extrapolating backwards to the zero dose map. And actually, something that I always found interesting, I don't know why people haven't looked more into that, because when I first saw that paper, I thought it was amazing, but no one's looked back at it too much more, even including Chris Russo and those guys who did it. So, but anyway, long story short, I think it is potentially a huge limitation for our current structures.
Okay, perfect. Thank you, Alan. I'm going to try to see if I can get a couple more questions in. The next question I have is, do you pre-process the 2015 Krios data using the new software that is available now?
It's a great question. I have not. But yeah, that was another huge tailwind that we had coming from the Krios to here. So I have not processed it, but Shores did. So, and Shores got that data set to 1.9 and maybe beyond. And actually, and with there was a couple obviously big improvements in the processing between 2015 and the later structure. Obviously, one was the Bayesian polishing, which is the Bayesian part was added in later. But also the big one was CTF refinement, which for myself, unfortunately, when Shores came out with CTF refinement, it proved that I had made quite a few mistakes in microscope alignment back in the day. And with the software that him and Yasenko developed, that fixed it. But so with the 2015, at least in Shores' hand, I know he got the resolution to 1.9, and then here with a similar image processing scheme, we got it to 1.8, and at that point, at 1.8, we could see hydrogens, which no one could see before in beta gal.
Wow, that's amazing. Okay, let me go to the next question here. Does the cryo breakthrough have to do with the sample fixation, or does it have to do with the electronics of the scope?
I think that, and again, so with this scope here, it's a 200 kV scope, and that's something to keep in mind. Also, an earlier generation of the CryoARM, but I think that the messages that I take home from our work and from other people's work in the field is that the specimen preparation is the key. So if you do everything right, you're going to get a high resolution structure, and that's kind of it. And I think that even I showed the slide at the end about the differences between the CryoARM 201 and the CryoARM 202, and the CryoARM 202, in theory, optically is better than what I have behind me. But I think that if I'm being honest with myself, if we had done those data sets on our CryoARM 202, I'm not really convinced it would have made much of a difference. I think that we have to do a better job with the specimen prep, and I think that's the main issue.
Okay, all right, thank you. All right, I'm going to squeeze another question in here. How do you transform the microscope data to generate these animations? Do you have a specific software recommendation?
For the animations, for these ones, I actually used old Chimera. I know that people like Chimera X, especially for visualization things, but for myself, I'm old and I'm used to regular Chimeras. And you just kind of apply the rotation, and I forget what the screen capture thing is, but for all the animations I showed, they're all done with regular Chimera.
Okay, thanks. Okay, and another question, let's get this one in. Can you elaborate on how you might have adjusted image collection settings to maintain high resolution as you progress from samples like ribosome to small proteins like HSA? And were these similar mag beam settings, exposure time, dose rates, CDS, and size and data sets, number of frames, etc.? I know it's a little lengthy there.
Oh yeah, it's good. Let me try to think. So the one question I remember as you were asking it, which is the difference, is the magnification. So the magnification that we used to arrive at some data set, the physical pixel size on the K3 was 0.52, and then for the smaller proteins that we do at high resolution, we run those at about 0.32 angstrom physical pixel size. For the other ones, so with CDS beam, those obviously for the beam settings is a bit tricky on the CryoARM 200 compared to the Cryos, or even compared to a CryoARM 300, because you have less flexibility in the lenses. So I actually don't know the beam settings off the top of my head, but you're a bit locked in because you can only have essentially one beam setting where you're parallel. You know, you can adjust the spot size and the angle and stuff, but you have to move in kind of discrete steps, and you have less flexibility with that than you have on a CryoARM 300, where you have the extra condenser lens, and certainly less flexibility than you'll have on a Cryos, where you have the free control. So I don't know those off the top of my head, but whatever they were, if they were different, it was completely by accident.
Thank you, Alan, for that. Okay, this is going to be our last question. This audience member is asking, Alan, how much improvement do you think is needed in sample prep technologies like plunge freeze to meet resolution challenges for both small and large molecules? Your last thoughts on those?
Yeah, it's a great question. It's the number one issue. We have a group here in the building that has the Vitrajet, and obviously more and more people are using things like the Vitrajet and Chameleon and seeing how it goes. And there's been quite a bit of work on that. I know Mark Herzig's group is showing some really good results on small proteins from these types of grid making devices. So I think that, yeah, that's one of the last remaining challenges here. Now that we, the microscopes look really good, the detectors look really good, and I think the specimen question is definitely the key challenge going forward.
Okay, thank you so much for that, Alan. Well, thank you very much for those answers. We have reached the end of the Q&A portion of the webinar, and if we couldn't attend to your questions, and there's a number of them out there, the team at JEOL may follow up with you after this presentation. And if you have any further questions, please direct them to the email address that you see there on your screen. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our webinars. Additionally, I will be sharing a link, so give me a few seconds to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. And for those of you watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing the recording and more. Now, please join us in thanking our speaker, Alan, for his time here today. Oh, and before I close out, we do have a link I'm going to put in the chat box right now. If you want any further information or resources, please utilize this link, and everyone should see it there now. There you go. All right. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X-Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Bye, everyone. Bye, Alan.
Bye.
Bye.